And before we start, we'll ask Dr. Habiba and Dr. Amina if we can have like a very quick session for questions from this morning presentations. Dr. Mina and Dr. I'm sorry for that. We couldn't have like questions in the morning because of the lunch. So please, if you have any questions about what our speakers they present this morning, go ahead.
Not the question. Actually, I already shared my thoughts with Habiba also. Conservation agriculture, which she presented, this is very of course, this is one of the major innovation. ICARDA just completed 50 years, just 5-6 days ago. And in our 50 years of experience, with working many countries, many partners, many places, conservation agriculture for dryland is the best solution anyway. Maybe we can combine with good crops, good varieties, stress tolerant in this one further, and then that can be better. And my expectation is maybe like in Morocco, then maybe some milestone there also might come. And then, here, you know, here in Morocco, we have 1 million hectare conservation agriculture by 2030. This is the, yes.
Thank you very much.
Okay.
Any other questions, please? After we finish with the questions, and so we have question over there.
Thank you. My question is for Madam Habiba. I just want to know how did you apply? I'm here. I just want to know how did you apply the 100 or the 3 water regimes of 150 and 30% of crop evapotranspiration, and then what are the water requirements of barley? And thank you.
Yeah, actually, we started with how much water the farmers are giving. Then we calculated using the calculation method for the water use efficiency, how much water is needed. Then we reduced that amount. So we use the 100 according to the farmers, how much they are using. Then we reduce it to 75, then 50%. Then we calculate it at the end, and we end up with the result, and now we are publishing it. Yeah. What is the other question?
In millimeter, like just.
Yeah, usually the farmers, they put 3 gallons per plant. 3 gallons per day per plant.
Like the amount in the whole phenological stage of barley, like the amount of.
But now we are reducing and we are calculating. So at the end, this is the paper. Now we will publish it.
Okay,
it's
thank you.
Finished already.
Thank you.
Thank you.
Thank you very much. Last question. So if you don't have any questions, I have a question for Dr. Mina. We said that you showed that residue can be good solution for salinity of soil. So we are working with our group, we are working in sites where there is like a high slope and there is a lot of topography. And do you think that the residue could be also part of the problem, because the residue, they will retain, they will keep the salinity from moving down or to other parts of the field? If we keep residue, can we increase the problem of salinity? Instead of having a leaching for salinity, we're going to keep it at the surface. Yeah, go ahead.
Actually, this was, not all the solution works everywhere. Yeah, this was mostly for irrigated drylands, flat land in Uzbekistan, where and then it is a bed planting. So bed planting, we don't recommend for the slopey land, because if there is slope, it doesn't work very well.
Okay.
Yeah. So it should be in the flat land, And then definitely in Morocco, I will not suggest to keep because it's really, really important, yeah, not that level. But Uzbekistan, where there is very highly intensive, you can imagine wheat productivity is like 8 ton, 9 ton is normal. So you know, you can imagine how much is the even stubble is very high, and then 2-3 crops. So, but here I will not recommend for slopey land. Yeah.
Okay, thank you very much. Yeah, go ahead.
Yeah, actually, I'd like to add something. In Kuwait, I established this breeding program because we didn't have like local variety for barley or barley wheat or sorghum. So I established this program, started from screening some varieties, then crossing, then after that applying the mutation on it. So this is, I started with my team. Then after that, that's why we decided to apply this all conservational agriculture to be sure is the variety is good for the farmers. And also we are working directly with the farmers. So our station is inside the farming area. Thank you.
Okay, thank you very much.
So can I have a question for?
Dr. Habiba, thanks, Dr. Mina. We can give them applause.
Yeah, can I have a question?
Yeah, go ahead.
So, if you
last
allow me, yes.
question.
Yeah, thank you. So my question is for Dr. Mina. So this has two components. Number one, in one of your slides, you indicated that the average yield of wheat here is around 2 ton hectare, and you are aiming to make it 2.8 ton. Is that not quite low? And if this is low, what are the reasons? Variety or something else? First. The second thing, because you are working to promote CA, what are the major challenges you are facing in the region which are hindering farmers' adaptation to CA practices? Thank you.
Yes.
Thank you. Yeah, it was you know, the year productivity depends upon the year. The year was, you know, the average yield, if you look in the Morocco, is around 2 ton. It's average yield. But the data we collected was somehow dry year. So this is only, not target is to reach 2.8, but we did that what are the best performing farmer are achieving this yield on that year, particular year. So it's like definitely target should be high, you know, at least 3-4 ton. This was only for that particular year, farmer were getting this yield because the year was very dry. And also another one is for conservation agriculture. What was? Okay. Yeah, exactly. So we have done a lot of comprehensive study on adaptation barrier. So there are many. Yeah. In Morocco, one is open grazing system to full apply the residue. Yeah, I mean to keep the residue. And another one is still, I will say, mindset of the people, you know, the farmer. Still, they are not so much getting aware. And another one is definitely the, you know, we need some kind of incentive. And first one was before, now we have a lot of machinery. Otherwise, zero till was very, very expensive. Now, Morocco has its own manufacturing, and also there are some other different options. Definitely one is machinery, I will say. Another one is mindset. Still hard to, you know, convince a farmer. We have to work. Now it's a good, this with 1 million hectare is really doing great, you know, working with the farmer. And another one is definitely the open grazing system. Yeah.
So thank you again. Thanks, Dr. Mina, Dr. Habiba. We're going to start our session. We are a little bit late, but we don't have any more session in this room, so we can, we will be okay. Yeah? So in this afternoon session, we have like 1 keynote and 3 presentations. So the first presentation from our keynote speaker is about crop suitability under open field and protected farming systems in arid dry land using APSIM and aqua crop models. And we have Dr. Krishna Devkota. Sorry for misspelling your name. The floor is yours. Go ahead, please.
Thank you so much. Good afternoon, colleagues. Myself is Krishna Devkota. I am senior scientist, ICARDA. I am agronomist and crop simulation modeler. I do now, my agronomy is around dryland farming system. In this next 20 minutes, I'm going to present about crop simulation model for decision making under diverse production environment in arid lands and Savanna region. Which one is next? Oh, okay. Okay, good. Many of you know this is Savanna region. And ICARDA, we work in Savanna region, which spreads from Morocco to goes to Kazakhstan. It covers mainly those 5 regions: North Africa, Nile Valley and Red Sea region, Arabian Peninsula, South and West Africa, and Central Asia and Caucasus region. We have somehow directly or indirectly, thinner or thicker, our presence in many of these countries. In this room, I was expecting a lot of students, but I am seeing very less. My point is, like, a crop simulation model is one of the very much required, need to understand. Very recently, I run wheat model in all countries. And I'm showing few of this result, how it looks. I'm sitting in Morocco, but I'm having very good idea of what is happening, what needs to be done. Yeah, okay, let me start my presentation with this. Anyway, total population in Savanna region, around 10% of the total population lives in this region. But water resource wise, this is the most scarce region. Only 2% of the total water is there. And this is known as water scarcity region, yes. Not moving. Where? Can you, why it is not moving? Okay, I was pressing another one. Thank you so much. Yeah, just I'll go very quickly. The challenges. Challenges are same. Everybody has understood. There is drought, there is rainfall variability, there is declining rainfall. Morocco is suffering from last 6th year now in a row, suffering from the drought. And this is not only in Morocco, among the many countries where we are working. Uzbekistan is also similarly affecting. By the way, this last part of the Uzbekistan, where there is a lot of farming, they are also suffering very much similarly. Then, second, of course, the major problem in the region is soil salinity. We know that 833 million hectare global salt affected area. Out of this, Savanna region covers 30%. Savanna is the major region which has been affected by the salinity. Around 300 million hectare is in Savanna region. And of course, insufficient irrigation and drainage practices, especially in Uzbekistan. I think in Pakistan also, problem of drainage, shallow drainage, or over-irrigation, secondary salinization. Those are the challenge in the irrigated drylands. And unsustainable crop production practices. This is one of the graphs we did recent survey in Egypt. If you see here, nitrogen use efficiency should be around at least 30 to 40%, but there is a lot of over application. Down from around 30% to down is over. Above there is, this is Egypt, they apply a lot of nitrogen. There is more over application. Similarly, phosphorus use efficiency also outside the range, it is either loss or soil mining region. Phosphorus is mostly in mining region. Similarly, this maybe that part, left hand side part, if it goes up, then it indicates mining, and down part more means there will be the loss. Similarly, labor, a huge number of labor they use. Similarly, other resources. This also creating big challenge, and all these challenges have compounded the risk of food security and demand for the urgent need for climate-smart agricultural practices, especially in the dryland countries where they need to rely heavily on water, and water is scarce. So here, how crop simulation model can help to make the decision in drylands. You know, conducting experiments is very expensive, limited by resources, people, maybe may not be possible to conduct many experiments over many locations, etc. And those kind of gaps can be filled up through the crop simulation modeling. Advanced crop simulation model have become decision support tool to researchers and policymakers. I use generally process-based model, upstream, DSAT, aquacrop. Those are the main model which I'm using, and also I'm showing the results. To predict crop performance under diverse conditions and develop adaptable climate-smart management practices in diverse crop production systems. Few examples where we can use simulation. For example, predicting sowing date. I will show few examples. Fertilizer rate, time, method, agronomic practices. Similarly, tillage, the agronomic practices means depth of sowing, distance of sowing, bed planting or furrow planting, direct seeding, many options are there. Climate change adaptation, actually modeling has been widely used in climate, predicting the impact of climate change. Similarly, yield gap analysis and carbon trading things and breeding support, nowadays this has been used. And then more extensively in G by E by M or C by E by M. I used to say C is crop and other are environment and management. Sometimes G is genotype. And to make sustainable practices also. So let me give a few examples. I'm just showing few examples. I'm not going through all these results. My point is how we can use model for different purpose. And of course, there is a link with salinity, irrigation water salinity, soil salinity, those things are coming. Yield gap, like this is the yield gap in MENA region. I run model for 12 countries here, from Morocco to Iran. You see those three bars are national average yield, attainable yield under rainfed system, and yield with supplementary irrigation. I didn't conduct experiment, but I used experimental data to make the model, and then I run the model for many years, starting from 1984 or something like this to 2024 or 23, up to now. And here, finding is also, I like that one. Actually, in rain-fed system, we say that we cannot increase yield. But even in rain-fed system, we have good potentiality to improve yield. This simulation shows that around 2.1 ton yield can be improved with the good agricultural practices, combining with good varieties, as I said, G by E by M approach. Similarly, I have quantified here the yield gap percentage. On an average, around 51% yield gap is there, but ranging from 36 to around 67% is in those countries. Now, I'm running in all C1 origin countries, which I saw, and then with many agronomic practices and combining varieties, etc. In future, it will come. Then how much water, if we go with supplementary irrigation, how much water we really need to apply? See, here all rainfed sites does not receive like very low rainfall like in Egypt. There are many sites that receives 400, 500 millimeter also in wheat season. I had seen one good question, how many millimeter water is required? We have all those answers here. Then, how many millimeter requires is around 96 millimeter on an average. If I talk about Morocco, at least if we give 2 supplementary irrigation, that crop yield can be increased by at least 20 or 30. I have quantified actually country-wise, long list is there. Here I will not be able to show. Just I'm giving example. Similarly, how crop simulation model will be helpful for the seeding date. Just I run last week the simulation for Tunisia. 4 sites, 5 sites here in that site, Beza and sites. And if you see, those are all our rainfed system. And see the yield variability. In one side, there is almost very close to 0. Other side, even 8 to 9 plant also is possible. See, but farmers are not getting. Those kind of decision is possible through simulation model. Then this I run the same in the same site. I applied the supplementary irrigation. How I applied is irrigate my crop when soil water content drops to 50% of available water, and then my yield increased. And here you see we are conducting experiment for many years to identify the seeding date. What's the seeding date? I was working in Uzbekistan 2 years ago. I didn't know what is the seeding date of wheat in Uzbekistan. Using model. It's very easy, very practical and reliable also if we use a model correctly, okay? Similarly, that's again the same reason with supplementary irrigation. See how much yield can be increased. This is the salinity effect in wheat. I run this model, by the way, the model which I run is APSIM-X. And I run the model for one of the sites in Tunisia, in the Beza site, and see yield decrease significantly after 9. This is Simon, up to 9, almost no change. Almost no change, this is there. Similarly, this is of course can be extensively used in climate change studies, and one of my students very recently did this job for 7 countries. We run the model for 7 countries. Yeah, I'll not read all names. Turkey and then Jordan, many wheat producing countries. Morocco of course is there, and there is the yield increase or decrease under different climate change scenarios. Yeah, this is there. Similarly, where we can use model is determining determinants of yield gap. We do not know, we know that this is the yield gap. We quantified using the model. And using the model again, we can quantify how much is the contribution from different production factors. How much is the contribution comes from variety, how much comes from conservation agriculture, how much comes from irrigation. Then we did this quantification, also possible. And we did for Morocco last year, and we run model for 3 sites. See, Massus, then Meknes, and see the LAD. And if you see these contributions, those are different. See, in irrigated site, one factor is important. Rain-fed site, another factor is important. As I said, I'm not going in the result now. This is because of the time. Similarly, all countries has the goal of food sufficiency, self-sufficiency. What is the effort countries are carrying to close this yield gap and make the country self-sufficiency? Very less. I would say honestly, very less effort is there. And then we tried that same exercise for Uzbekistan. We run again the model. And then 3-4 sites, Khorezm, Kaskadaria, Samarkand, Jikha. And then we quantified, see, the response of water, the response of fertilizer. Using model again, we quantified how much water we need to apply, how much fertilizer we need to apply, possible. See, and then we run that one using this model, we run machine learning as well. And again, we validated our results using machine learning. That graph is from using machine learning. And this has just been published also. So here is crop molding offers a science-based framework for improving crop productivity through agroecological specific climate social system. Now, very recently, this is date palm. Date palm is major crop in dryland region. We all know that one. What are the efforts we are doing? There are a lot of challenges, also problems in date palm also. What are the efforts we are doing? Do we have understood its growth? Well understood? Actually, when I started to make this model, I encountered a lot of problems. There's no data. This is perennial crop. Of course, we know that getting data is difficult. But anyway, I'll not go in this story now. But recently, we don't have any crop simulation model for date palm. And I myself make date palm model and APSIM-X, and this is available, and I run it for 182 farmers field in using data from Abu Dhabi, real soil, their own climate, and this is the graph of the yield when FFP is the fruit yield. Then it is ranging. I have actually made the model for 2 crops, and again here also it's possible to give what is the fertilizer rate for date palm for farmer wise. What is the plant when they should irrigate? Actually, I have seen date palm next to rice. They are putting so much of water in date palm. Is it really necessary to apply that much of water in date palm? No, that's not necessary. You see, around after 30. If the soil water content goes below 30% available water, yield is decreased significantly. But farmers are putting 100% saturation condition, and they are putting 3,000 cubic millimeter water in 1 year. This is the reality. APSIM and eco crop, I have actually made both model for date palm. Then another exercise, I'm almost in a few slides. Another exercise, big exercise, we did for Abu Dhabi. What we did is 51 crop simulation under 182 farmers field under 3 production systems: open field, greenhouse, shade house. Farmers had greenhouse previously there, and greenhouses are temperature high, and then nowadays they are moving towards shade house. What is the yield difference? What is the attainable yield? Where is it? And there is a lot of issue of soil salinity, irrigation, water, etc. is there. And out of 51, 18 were vegetable, 9 fruits, 9 forage, 5 food crops, and 3 production system, and 182 farms. I used 7 APSIM as well as crop cyst for 8 crops APSIM, and for 43 crops we used aqua crop. And this you saw in the that is the book FAO is publishing now for all 51 crops. See, I will not go in this table now, but which I explained already. And out of 51 crops, I had to make new model for at least for 27. Actually, that was big work, at least 15 people were working. And those are, I will not read this result, but if you see, just I will explain from one. Pumpkin. See, there is attainable yield, and here is the PY is potential yield. Big difference from here to here. What is the factor for this one? Is 3. In this condition, of course, salinity, soil salinity, and water salinity, and then drought and heat. We have quantified all those effects as well. Here just I'm scrolling down. Those are the any perennial forage. Actually, forage study is very less, especially in simulation also. And why I'm doing this exercise also is we have a lot of crops opportunities. And we know that this contains more nutrient, that etc. But in terms of agronomy, very less has been understood. So this needs to be a bit explored, especially by the young students. Food crops, I will not go. By the way, quinoa, I'm very happy. This is attainable yield is almost very close to potential in quinoa. This is good crop compared to other crops. Similarly, there is forage. Yeah, everything is coming in few months. Here we try to quantify the effect of irrigation water salinity on those crops. See, of course, our interest is around up to around 20 deci because this is the area where must we grow crop. So see many crops and all yield is decreasing, and then this is the effect of irrigation water salinity. Then here is the soil salinity effect. Similarly, for all those crops, what was the reason? Then my point here is science-based agricultural planning for crop suitability. Actually, that was used for crop suitability mapping of the region. Which region is the good for which crop? And this approach actually is, I would say, replicable and scalable. And we used it very recently to make the master plan of UAE. Then, considering the need, actually, that's the reason I was saying we need more crop simulation modular in the region. Last year, that was 2022-23 February, we have launched AgMIP MENA. Actually, ICARDA and UM6P has launched AgMIP MENA. What is AgMIP? AgMIP is Agricultural Model Intercomparison Project. This is international project. This is an international channel, and this is managed is Alex Raune. And yeah, this has been started. And our objective here is to assist, of course, the climate change impact in agriculture at different scale. Then provide decent support to the farmers and policymakers. Actually, this is very useful. And to understand what is the research gap, it's a very good tool. Then train and capacity building up crop simulation modular, and fourth is to create database of crop, soil, climate data set, which is severely lacking. And if I try to find, it's very hard. Everybody says I have, but at the end, I will not get anything. So those are the objective, and maybe in future we'll organize some sort of online training or something like this in crop simulation modeling. This is required, looks like. And conclusion is, anyway, crop simulation is the data-driven decision support tool. Science-based agricultural planning, as I said, we validated our work. Several rounds of meeting with national partner of how many scientists, and everybody has agreed this is fine. And development of agriculture master plan of UAE, simulation model, and that can be used for many other countries. We are thinking also maybe we will use the similar approach for other countries also using simulation model for the agriculture plan, development of plan. And the simulation modeling actually is the blueprint for the suitability mapping and evidence-based crop zoning and investment decisions. Many examples are there. Yeah, it develops a localized adaptation to landscape level. It's possible, a lot of things are possible. And to quantify the effect of climate, management practices, drought, salinity, we are doing many things. And for sure, one model is not sufficient. Sometimes, as I said, we used aquacrop because aquacrop has good soil salinity module. That's the reason. Sometimes we use hydros based on the need, okay? And actually, this approach, I would say, is replicable and scalable, especially for to design climate-smart agriculture to improve the food security. And I show that I have used for quinoa as well. And maybe next page, maybe I can quantify what is the planting rate of quinoa, which region, how much fertilizer it requires, many things possible. Thanks so much.
Thank you, Dr. Krishna, for this impressive presentation. Do we have questions for Dr. Krishna? Yes, please. You can keep it.
Thank you. Maybe I can give you my.
It's working, it's working.
Thank you very much for your nice presentation, Krishna. So my question is regarding the yield gap. So this yield gap is due to lack of genetic gain, good agriculture practices, or a seed system? Because you have extensive work in ICARDA while interacting with the farmers.
Exactly, this is a good question. As I said, I do not trust only in one factor, only one reason. You see, of course, there is the interaction of many factors. I would say even socioeconomics also affects. I say that, let's say, for example, I say you can increase your yield by 2 tons, for example. But there are many factors. Farmer may not have irrigation facility. If no irrigation, and you will be going down by at least by 30% of the total gap which he wants to close. Similarly, if he is not finding good variety, again another 25-30% will go down. So, you know, I was trying to make this 100%, and then decomposing that 100% to different factors. And then that's how we did for Morocco and then Uzbekistan, 2 papers. We have somehow quantified. And that approach can be used. We are trying to do similar approach for other countries also. But not only one factor, there are many factors. Okay. Yes.
Next.
Yeah, thank you so much, Dr. Krishna, for this nice presentation. So my question is regarding the date palm yield. As I say there, it looks like 80 kilogram of nitrogen per hectare is the optimal, let's say, nitrogen rate for date palm growth. I mean, for the optimal yield. Mm-hmm. But it looks for me a little bit high because if you look at the distribution of this date palm in Morocco, you will find it more in oases, you know, in the Asiatic area, especially in the southeast of Morocco, Rashidia regions, Zagora, etc. And I found this is a little bit, it should be a little bit lower. What do you think?
It is lower, yes. Lower or higher, you mean?
Normally, what you show is a little bit higher because 80 kilogram per hectare is a little bit higher.
You know, for UAE condition, I was thinking this is a bit low. I was thinking to make my model a little bit improve. Oh, yeah. Maybe I'm doing this exercise for Morocco also very soon. I have parameterized variety for Morocco, other major date palm producing countries. Yeah. And then I'm coming with that one. We will validate with this how much is the common practice and how much is the yield. We will match. And then country-wise, we will be making this cultivar. That's what I did not show many things actually. Here, to make model, I said model, but to make that one, it takes time and we need calibration, validation, a lot of steps are there. Yeah. Here, just I wanted to give some idea, model can do something.
Yeah. Thank you so much.
Good point. Good
that's
point.
Interesting.
I will note it, and
Thanks
when I run the model, I will see how much will be the optimal for Morocco or our region.
Perfect.
Good point.
Thank you.
Thank you.
So we have last question. Butaina, go ahead, please.
Thank you so much, Dr. Krishna, for your presentation. My question is it possible to have a predicted data, like predicted yield or something like that, using only data, weather data from 1 year? If it is not possible, what is the minimum data we can have to have a good prediction?
I would say season, not year. Let's say our wheat planting in Morocco, for example, it starts in November. And if you have data from November to June, July, up to harvest, it is fine. You can run even with single season weather data also. This is a good point. Actually, this is seriously lacking in the region. Data availability. Currently, we are trying to make a soil and climate data repository so that we can provide that to our modulary who are working in the region. This is, good point, but possible. Yeah.
Thank you so much.
Yeah.
So
Professor.
Thank you, Professor Krishna.
One
Okay.
question? Yeah.
I said
Yeah.
last, but
Yeah.
Thank you very much, Krishna. You covered a large area of, but I will ask you about one of the slides that you mentioned on the date palm and the crop water requirement of date palm in Emirates, where I came
Right.
from the same area. I don't know whether the program that you, or the model that you used, have you estimate the actual crop or date palm water requirement, and have you added the saline or the salinity of the water that you're using, and what is the number that you got? Let me compare it with what I have as I'm working in the same subject. Thank you very much.
I need one separate whole list, Professor, for that one. I can show you. Actually, I have made model, a dead palm model using two models. One is APSIM, another is INDISAT. And in APSIM, I am running a salinity effect so far for many other crops, but it's still not for dead palm. That's the reason I again made model in eco-crop. And my model in eco-crop quantifies all those things, which you say, as you said. And all model, all crops, which I'm saying, especially for UAE condition, all inside there is measured data, there is management practices that farmers are really using. There is nothing hypothetical. We use the data which we use to make the for the UAE master plan. We have run.
Do you have a number of per tree, per hectare, per 100 tree of using the APSIM model?
That I can make both way. Now when I make the model, I have put either 135 or 156, 7 by 7 or 8 by 8. That's the planting geometry they use. That I have both way. It is possible per hectare, per plant, and dead palm generally I find data in per plant basis, so per plant is also there. Like this, even this FFT which I showed, in another graph I didn't show, there is a per plant, per tree also. This is a good point, and actually in all those 51 crops, there is big effect of soil salinity in these dry land countries, and big yield gap has been mainly affected or mainly due to soil or irrigation water salinity. Yeah.
So I think the time is limited. We invite you to keep
please.
discussing together.
Okay.
Thank you again, Dr. Krishna, for your nice presentation. And now I invite Mr. Abdul Hamid from the University of Karachi, Pakistan, to present his work. The floor is yours. You have 12 minutes, please.
Assalamu alaikum. Good afternoon. I am Abdul Hamid, Associate Professor at the Institute of Sustainable Halophyte Utilization, University of Karachi. I am a plant physiologist, mostly focusing on the salinity tolerance mechanisms of halophytes, particularly their seed biology. And today I will be sharing with you some of our recent research on seed enhancement of some of the cash crop halophytes. As we are all aware that the world population is increasing fast, and by 2050 we will be around 10 billion people, which means world would need about twice as much food as we need today. It also means that we will need about twice as much water as well as arable land for farming, for achieving food security by that time. But unfortunately for many countries, especially those which are in the arid region, including Pakistan, where I belong, we don't have many options for farming more land with more water. For instance, in 1947, when Pakistan was established, per capita water availability was around 500,000. And by 2025, we are at the verge of becoming a water scarce country. In addition, the subsoil water in the arid region is also not recharged with fresh water. Generally, it is of poor quality and not fit for the most plant and human consumption. In addition, the global climate changes, they are also hampering the food production, and according to some estimates, by 2040, the global warming will heat up the growing season enough to affect the crop productivity. So together, all these factors, that is intensive agriculture, lack of fresh water, global climate changes, they are leading to another problem of which is salinization of land. Today, around 800 million hectares of the land is already saline, and this number is soaring high with time, and by 2050, according to some estimates, we are going to lose 50% of our farmlands to salinity. And unfortunately, most of our conventional crops, they lie in this region. I mean, they are kind of salt sensitive, and they show signs of stress at around 4 decisemen salinity, which is around 40 millimolar salinity, and the salinity in many of the arid areas, especially which are close to the coastal regions, it is very, very high. Sometimes it is as high as seawater salinity. So, and opening new land for cultivation is also not without dire ecological consequences, while finding additional fresh water in our part of the world is also a remote possibility. So what is the solution? We need development of salt-resistant varieties that can take agriculture to the marginal area to achieve food security by 2050. But unfortunately, salinity tolerance mechanism is kind of polygenic, and so far we don't have that much success in developing real salt-tolerant varieties of conventional crops, so that we can grow them in like coastal areas with seawater, which is plentifully available. So we believe, meanwhile, we'll see some real salt-tolerant crop varieties. Halophytes can be a good option, which is both ecologically as well as economically feasible. These are a small but diverse group of flowering plants, which are capable of completing their life cycles in habitats with at least 200 millimolar NaCl salinity equivalent. They are kind of an untapped resource. They can not only use for improving salinity tolerance crop by serving as model, of course, but they can also be utilized as non-conventional crop for the saline lands. Halophytes, a number of halophytes, of course, they have a number of economic usages, such as they can be used as feedstock for biofuel production, they can be used as food and for the edible oil production, as fodder, for research, industrial, they can provide industrial raw material, landscaping as an ornamental plant, they can be used for timber formation, as well as they can be a natural source for various chemicals. A large number of halophytes called euhalophytes, they show very good salt tolerance. They are capable of surviving as high as seawater salinity, and some of these plants, for instance, Soida fruticosa, which is common in around Karachi coast and many other parts of the world. This plant shows kind of stimulation, growth stimulation in 300 millimolar salinity or even 600 millimolar and even salinity, which is equivalent to seawater salinity. Similarly, there are some other plants which show salt stimulation and they can survive very high salinity. And they do so by employing a number of morphological and physiological adaptations, which I'm not going to discuss today. Focus of my research is on their seed germination. When it comes to seed germination, halophytes, they are also kind of sensitive. A number of halophytes which are highly resilient to salinity at growth stage, they show some inhibition of germination. For instance, Soida fruticosa, that can survive about twice as much as seawater salinity, is highly sensitive even at 300 or 400 millimolar salinity. So most of the halophytes which are highly resilient at growth stage and have very good economic potential, they also show some inhibition during when it comes to germination, which is their first stage of their life cycle. So in this context, if we want to cultivate halophytes at mass scale, we have to have ways to enhance their seed germination, so that they can be recruit good population in the under highly saline conditions. So recently we started working on this seed priming, which is a kind of physiological enhancement of seeds. We soak seeds in water or any appropriate solution for a certain period of time, then dry them and then sow them and expose them to various stress conditions. And most of the time, the prime seeds, they do better, they perform better than the unprimed seeds. And there are a number of priming techniques depending upon what kind of soaking solution you are using, like hydropriming, osmopriming, biopriming, and redox priming. We tested a number of priming techniques and found that this redox priming is kind of better for the halophytes which are found in our area around Karachi coast. So redox priming is soaking seeds or priming seeds in redox active compounds, and these compounds mostly serve as like kind of redox signals, and they activate various resistance mechanisms in seeds so that the seeds which are primed, they perform better under stressful conditions. Objective of our research has been to examine efficacy of various redox priming procedure on seed germination and salinity tolerance of common local halophyte with crop potentials. This is a very brief outline of the methodology that we use. We soak seeds in redox solution for a certain period of time, and of course, we optimize the duration as well as concentration of the priming agent through preliminary experiments, and then we perform, germination and seedling growth, and and also look for the mechanism under both saline and non-saline condition. This research was done on two halophytes, Zygophyllum simplex, which is a medicinal halophyte, has a lot of potential for development as a medicinal plant, and Uroconda setulosa, which is highly salt resistant, found in the subhas with very high salinity, and is used by the locals for as the forage grass. You can see in case of Zygophyllum simplex, seeds, they have dormancy. For example, look at the 0 millimolar bars. The germination was very, very low. It means they have like innate dormancy. And when we prime the seeds, the dormancy was alleviated, and also under saline condition, all the prime seeds, they perform better than the unprimed seed. In case of Uroconda setulosa, there was like no dormancy, and there was no difference under prime and unprimed treatment. But when we look at the germination under saline condition, on most of the instances, prime seeds, they perform better than the unprimed seeds under increasing salinity. Not only seed germination was okay. Okay, not only seed germination was enhanced, seedling growth was also stimulated. We also performed, this experiment on melatonin, which is another antioxidant, and we found some good result. On most of the occasion, the seeds which were primed with the melatonin, they performed better than the unprimed seeds. Seedling growth was also promoted, and so were different physiological parameters. So in general, in totality, seed priming can be used to overcome dormancy, which is a common problem in a number of halophytes, as well as it can also stimulate their germination under saline as well as non-saline condition. We were also interested in how different redox compounds are, leading to the same stimulation, better results. So we wanted to look into some of the mechanisms. So if we analyzed these signaling, compounds, hydrogen peroxide, nitric oxide, and ascorbic acid. Under non-saline condition, the levels of these redox compound were like kind of similar between prime and unprime, but under saline condition, all the prime seeds, they showed relatively higher levels of these signaling compound, H2O2, nitric oxide, and ascorbic acid. So we wanted to know which one is important, or whether they work in a kind of, they work together. So we performed this pharmacological experiment. We employed potassium iodide and sodium pyruvate, which are the quenchers of hydrogen peroxide. Then we supplemented it with nitric oxide, which is SNP, which is a donor of nitric oxide. And we also supplemented in some of the treatment with ascorbic acid. So when we quenched hydrogen peroxide, like the germination was reduced. And when we supplemented it, this potassium H2O2 quenching with SNP, that is exogenous nitric oxide, there was some reversal in the germination. But that did not happen in case when we supplemented H2O2 quenching with ascorbic acid. It means that the H2O2 work in tandem with nitric oxide, but not with the ascorbic acid. So, so we believe that the H2O2 and nitric oxide, they provide kind of signaling for the improvement of germination when we prime seeds. So in short, redox priming can be used as an effective strategy to enhance seed germination of halophyte under stressful condition, and probably nitric oxide mediated RO signaling is responsible for this, improved. In future, we are interested in studying the downstream molecular targets of this signaling, whether there are a carryover of the priming memory to the next generation, and what are the molecular bases of priming memory. These are some questions which we want to, discuss in the future. We obtained funding for this research from University of Karachi, Higher Education Commission of Pakistan, and Sindh Higher Education Commission. And I would also like to take this opportunity to thank the organizers of this conference to invite me and providing the opportunity to share my results with you. Thank you very much.
Thank you so much, Professor Abdelhamid, for the presentation. And I'm asking the people if you are having any question for Professor Abdelhamid. Anyone? So maybe now. So let's go to our next presentation by Professor Xavier Yusius Garcia from University of Zaragoza, Spain, who will be presenting self-rooted hazelnut cultivars under salt stress. Thank you. The floor is yours.
Will you pass the slides? Good afternoon. Thank you for the invitation. In reality, I am not a professor. I am a PhD student. A little bit old, but I am still a PhD student. No, it's, I am going to, this paper is already published in Agronomy magazine. The idea is that we develop a a paper regarding hazelnuts. Hazelnuts production is based in Turkey. There is a big demand. Companies like Ferrero Rocher is lacking reliable production of hazelnuts, and hazelnuts is expanding to new countries like Chile, South Africa, Australia, with moving to marginal lands, and the idea was to analyze the resistance of 4 not root stocks, genotypes, because hazelnuts, they are self-rooted crop, and compare with the 2 Tonda Gifoni and Tonda Romana with 2 American cultivars, Yamhill and Barcelona, regarding to salinity. We know the production, we know the yield, we know how it behaves, but really there is a lot of lack of information in hazelnut crop regarding to salinities. And well, the idea was to characterize the different resistance to be able to give advice to the growers for the new plantings, especially in these new areas. No? It was done in a greenhouse in the University of Zaragoza. The trial was run by 5 months. It was in plastic pots. It was sandy soil or sand. We were using 1/4 of Arnold solution, and we apply 4 different sodium chlorine solutions. And with the 4 cultivars, we were irrigating to have extra water as a leaching to be sure that the salinity in the pots maintain the same values. And every 15 days we were measuring different parameters like leaf chlorophyll, chlorophyll fluorescence, and stomatal conductance. And after we remove the plant from the pot, we separate roots, wood, new shoot, and leaf. We did dry and fresh weight, and also we analyzed the cations and chlorine of in each organ to know the accumulation of sodium, chlorine, calcium, potassium in the different organs. As the fresh and dry weight, it was clear that Tonda Gifoni and Yamhill maintained the better fresh weights across all the salinity treatments, and roots and wood demonstrate more resistance to salinity. Regarding chlorophyll fluorescence, Tonda Gifoni again maintained better photosynthetic function and electron transport, and between 50 and 75 millimoles of sodium chlorine causing severe damage except on Tonda Gifoni. As visual symptoms, this is at the end of the treatment. At 75 millimoles, all the plants were dead. At 50, some were maintaining some leaves. Really, it was at 25 millimoles sodium chlorine that it was showing some difference. And Barcelona at 50 maintained a more foliage than the others, but at 75 all showing a severe defoliation. The root system in Tonda Gifoni was much dense, much more compact. After Yamhill and Tonda Gentile Romana as showing as more accumulation of chlorine and sodium, the root system was much more small. And we were also measuring the damage in the leaves in a 0 to 5 scale, and we were indicating when the first symptoms appear for the different scale and when the plant really died in the 13 fortnights during the 5 months that the experiment it lasts. In the cation control, it was clear that the roots maintained the highest calcium concentration through the experiment, and leaves maintained the significant reduction, and wood and stems, it was more like a storage. In potassium, Barcelona exhibited the most substantial reduction in the higher level of salinity. and leaves maintain the highest potassium concentration through all the experiment. And again, steams and wood exhibited important losses of potassium. In chloric content, at 75 Thunder Romana showed the highest concentration, that it was the most damaged genotype, and leaf, as normal, increased the concentration of chlorine across all the salinity treatments. In sodium, follow a similar pattern with leaves more increasing the sodium content, and Tonda giffoni maintain the lowest concentration at salinity levels, and Tonda gentile romana showing the more higher concentration of sodium. No? With the ratios, potassium, sodium, in Tonda giffoni, higher ratio. This means that more potassium, less sodium, and roots and wood also exhibit more severe ionic imbalance and the high salinity conditions. And and in calcium, the same. Giffoni maintain the higher ratios. And a little bit of a conclusion. It was nice to see that Tonda giffoni, it has more vigor, more resistant to salinity, probably according to the bigger root system. Barcelona and Bianchi performed an intermediate situation regarding to the salinity resistance, and Tonda gentile romana, really we cannot recommend it in saline soils because it was really affected very quickly and very, very pronounced. And also it was clear that 50 millimoles of chlorine, it was probably the value that from there we are getting severe damage. And there is for sure that now we need to start doing this trial in the field to prove that the relationship between dry land and lack of irrigation and salinity, but at least is the first step to start recommending some of these genotypes for these new plantings. As again, it's already published in agronomy, and if you want more information, you can get the paper. Thank you.
Thank you so much, future Dr. Xavier. And I will ask all the participants if anyone has any question for Mr. Xavier. Can you please join me? Yeah.
It's not my specialties, but I do have a question for you. I noticed that all of your work is on seedling or very young trees or genotypes. Yeah. I don't know how long it will take to produce the hazelnuts fruits that it is, you know, you're gonna.
These plants, they are 2 years old. They were 2 years old. We prune it to 60 centimeters. We wait that the new growth should start, and we will start measuring the.
Yeah, I noticed that you start to measure, but how long it will take in order to have a production of the hazelnuts fruits, and then what, you know, what will happen with the mature trees?
With the mature, what is clear that with 50 millimoles of chlorine, sodium chlorine is around 6 decisiemens per meter of electrical conductivity of water. We cannot plant hazelnuts because the trees will die. I will take some risk in a sandy soils if I have extra water with a conductivity of 3 decisiemens per meter, but above 3 and a SAR of above 15, I will not recommend to plant hazelnuts because the trees will die in the next year or couple of years. A part of management, irrigation, mounding, gypsum, organic matter, but above 3 decisiemens per meter of irrigation water, I don't think that it's recommended to plant hazelnuts.
Even with the old one, you know, with the mature trees.
Yes. Ferrero Rocher did an important project in Australia. They planted more than 500 hectares, and after they didn't check the soil before planting, and after 4 years, they pulled out all the orchard. They have lost more than $20 million in Australia, and now we are going to plant olives because the soil has high content of saline, heavy soil, and the hazelnuts didn't cope with that.
Thank you.
Thank you so much. Do we have another question? Okay, so you're welcome. So let's move to our last presentation by Mrs. Hasna Zine from King Abdullah University for Science and Technology.
Well, hi everyone. I am Hasna Zine, a PhD student officially enrolled in Sultan Moulay Slimane University in Beni Mellal, but my PhD work is kind of collaboration between USMS, King Abdullah University of Science and Technology in Saudi Arabia, and Valoris in France. So in this presentation, I'm going to show part of my work, which is titled Seasonal and Diurnal Fluctuations in the Metabolome of Haloxylon salicornicum in a Desert Environment. So as we all know, deserts are arid regions characterized by an aridity index of 0.05. They are also marked by extreme temperatures, low precipitation, often less than 250 millimeters a year, sparse vegetation. These areas occupy 1/4 to 1/3 of the global surface, and this surface is increasing with the rise of desertification. A prominent example of such environment is the Arabian Desert, which spans much of the Arabian Peninsula and exhibits all these defining characteristics, including low precipitation and extreme temperatures. Despite the harsh conditions prevailing in the Arabian Desert, the flora remains quite diverse, including both ephemeral and perennial plants. Specifically, ephemeral plants survive harsh conditions as seeds and germinate when the conditions become favorable. In contrast, perennial plants survive over the year by relying on advanced morphological, anatomical, and physiological adaptations. Haloxylon salicornicum is one such perennial plant widespread across the Arabian Desert, and it is xerophyte shrub belonging to the family of Chenopodiaceae, with leaves reduced to small swollen tips at the ends of the stem segments. Its green stem takes over the role of photosynthesis. It is also characterized by a deep root system that can extend up to 10 meters. At the anatomical level, this species exhibits adaptations such as a thick cuticle, sunken stomata, multilayered epidermis, small vascular bundles, and it is also characterized by Kranz anatomy, which is a characteristic photosynthetic, which is a characteristic of C4 photosynthesis, where plants concentrate CO2 in the mesophyll cells and then pump it to the bundle sheet cells, allowing them to use a low internal CO2 concentration more efficiently. So anatomical and morphological adaptations of this species were widely investigated. However, little is known about its metabolism under natural conditions. So for that, in this study, we aimed to unravel the seasonal and diurnal fluctuations in the metabolism in the metabolome of Haloxylon salicornicum in response to desert conditions. So this study was carried out in the Shar'an Nature Reserve in Al Ula in Saudi Arabia, where we selected a sandy natural site devoid of any human activity and where haloxylon plants thrive. So here I'm going to briefly go over the assessed parameters. So we first assessed the soil elemental composition, and we measured soil pH. Both analyses were performed on soil samples collected from a depth of 40 centimeters, and we also measured the soil moisture at 1 meter depth. At the plant level, we measured the plant's water potential at dawn to see the plant's water status when it is in equilibrium with the soil water potential, and at midday to determine the plant's water status when it is under the transpiratory demand of the atmosphere. Simultaneously, we collected green stems to assess the primary metabolites using a targeted metabolomic approach. We assessed soil moisture, plant's water potential, and primary metabolites in both winter and summer. So the results showed that the soil pH was alkaline and the predominant element was silicon, which is an essential building block of sand. And due to the sandy texture of the soil, the water retention capacity was very low, leading to lower soil moisture values reaching as low as 1% even in winter. As for the plant's hydric status, the comparison between dawn and midday across seasons showed lower values at midday, which is the response to the high vapor pressure deficit. In contrast, the comparison between winter and summer exhibited low water potential values in summer, which indicates that the haloxylon plants were completely dehydrated because of dry soil and atmosphere. Regarding the metabolomic data, out of 5,000 peaks, only 86 peaks were identified using COVAD library. These metabolites were categorized into 4 chemical groups: amino acids and their derivatives, comprising 41% of the metabolites, followed by carbohydrates and their conjugates at 37%, and then carboxylic acids at 20%, while fatty acids were detected in minor amounts. Well, the differences between conditions were visualized using PCA, in which we can clearly see that there is a significant difference between dawn and midday across seasons, and this indicates that the plant's adaptive response to diurnal changes. And also there is a significant difference between winter and summer, reflecting the differences in the metabolic adaptation of Haloxylon salicornicum to different climatic conditions across seasons. To get a clearer picture about the relative abundance of metabolites, we performed a hierarchical clustering analysis, and starting with carbohydrates, in both winter and summer, there is a significant metabolic activity at dawn compared to midday, which is tightly linked to the plant's water status, which was moderately high at dawn compared to midday. Well, in winter, the concentration of glucose, which is the end product of photosynthesis, increased. This indicates that photosynthesis occurs during the day, as known in C4 plants. In addition, mannitol, sugar alcohols like mannitol, arabinitol, and sorbitol increased at dawn. These metabolites could contribute to plant osmoregulation process. When we look at the metabolites accumulated in summer, we find that approximately all of them resulted from starch degradation, either by hydrolytic pathway or phosphorylytic pathway. Similar tendency was observed for amino acid, and the metabolic activity was more pronounced at night. In winter, the abundance of valine, isoleucine, and valine, isoleucine, lysine, and glycine may contribute to plant osmoregulation as well as protein biosynthesis. However, dopamine, and GABA may function as signaling molecules triggering stomatal closure at midday. In summer, the most enriched pathways were tryptophan and glutathione metabolism. For instance, tryptamine and serotonin are known to have an antioxidant metabolic activity. An antioxidant activity under drought. However, indole acetic acid is involved in root growth. In addition, glutamate and cysteine glycine are two essential components for glutathione regeneration, which is a ROS scavenger. Sorry. Well, the last heat map shows the relative abundance of carboxylic acid, hydroxycinnamate, and fatty acids. All these metabolites showed significant metabolic activity at dawn. In winter, the haloxylon plants found to redirect their metabolism towards anabolic processes such as flavonoid and fatty acid biosynthesis, as shikimate, tartaric acid, and malonate were upregulated. In summer, the TSA cycle intermediates like malate, isocitrate, aconitate, and succinate were upregulated, ensuring continuous ATP production. However, coumarate, caffeic acid, pipicolate, and palmitate have been shown to act as antioxidant molecules under drought. Lastly, oxalate is an alternative source of carbon under drought. So to summarize, haloxylon salicornicum plants exhibited a significant nocturnal activity. This is likely due to the existence of robust circadian rhythm that enabled the plants to carry out their metabolism away from intense sunlight and heat during the day. Upon comparing the metabolic activity between winter and summer, we found that the plant's water status was tightly linked to the plant's metabolism, such that a high water status stimulated anabolic processes such as photosynthesis, fatty acid, and flavonoid biosynthesis. In contrast, low water status enabled the catabolic pathways such as starch degradation and mobilization of antioxidant molecules such as ascorbate, serotonin, tryptamine, caffeic acid, and coumarate. So this work partially uncovered the metabolic adaptations of haloxylon salicornicum to desert conditions, laying the groundwork for understanding physiological adaptations of desert plants. And thank you.
Thank you so much, Hasna, for your presentation. And since we still have some time, I would like to invite Dr. Krishna and Professor Abdul Hamid for any further question. Also, you as well, Hasna. Yeah, yeah. So, anyone, do you have any question for the three presenters here with us? Yes, Dr. Shaheen.
Thank you. My question is about the haloxylon salicornicum. You have done beautiful study and a lot of work you have done. What is the impact of such study in terms of food security or in terms of restoration of deserts? What particularly, your emphasis on?
It is. Is it on? Can you hear me?
Yes.
Yeah, okay, perfect. Actually, it's not, it's an explorative study to see what is going on in this plant under desert conditions. We cannot use haloxylon salicornicum for food security, but it is a promising species for restoring degraded lands.
Yeah, it is available in the coastal areas.
Yeah.
Very highly saline areas.
Yeah, it is xerophyte actually.
And what impact your study will have on the growth of Haloxylon salicornicum, or I mean?
It's actually just an explorative study of the metabolic adaptation of this species under desert conditions.
Okay, thank you.
Thank you very much for your presentation.
You're welcome.
I'm just wondering, when you measure the water status by water potential, is it the one in the dawn on the early morning? Is it after the sunshine or before the sunshine?
No, it's actually before sunset.
Before
the sunrise, actually.
Before sunrise.
Yeah.
Okay. And you get a -2 bar for
minus 1.5 megapascal.
That's very low. That's very good one.
Yeah.
Thank you.
Yeah.
Do we have any other question for our presenters here? Okay, again, Dr. Shane.
The question is for Dr. Abdul Hamid. Dr. Sahab, you have done a lot of work on halophytes, and halophytes are salt-tolerant crops, They can tolerate, they have the mechanism, Is there any way that we can induce such character into glycophytes through biotechnology innovations? Is there any initial work done on this in this aspect, or people are still thinking, biotechnology still thinking if this can be done as well? Because most of these sensitive crops like glycophyte, like vegetables, they are very sensitive to salt tolerance, right? So they are grown under greenhouses where a lot of energy is required, a lot of water is required. Normal farmer cannot afford such high expensive investment, So are we thinking of really adding some sort of salt tolerant gene or mechanism into these sensitive crops so that the farmer, they can grow in the open field with harsh environment and using saline water? Thank you.
All right. So yeah, there are many groups who are actually working on, producing transgene, from the different halophytic genes, right? But and they do report, promotion in the salinity tolerance of those transgenic lines. But our group is mainly focusing on the physiology of halophytes, and we believe that both halophytes as well as glycophytic, even sensitive crop plant, they have kind of similar salinity tolerance mechanisms. You see, for instance, both glycophytes and halophytes, they both have like catalase, and they have like those SOS pathway, and all the antiporters, you name it. All the genes which are found in halophytes and performing those resistance activity are actually also found in all the crop plants, including Arabidopsis thaliana. But we see the difference is in the level of regulation. So halophyte, they have signaling mechanisms, when they are exposed to salinity. So they use different signals to activate those genes for a very high level. And some of the halophytes, even they show a kind of constitutive response. So they just keep producing different resistant proteins and protective compounds even when there is no stress. And when they are exposed to stress, so they are already prepared. But for the sensitive crop plants, right, when they are exposed to stress and they close their stomata, and they are not prepared. So it's very kind of too late for them to produce that defensive machinery. So what I believe that studying the regulatory mechanism, signaling mechanism that are operative in halophytes, but are kind of missing in the glycophyte, that can have kind of lasting impact. And but I'm not aware that if any of the molecular research for this kind of deciphering these signaling mechanism has been done. And if we know the signaling in that is operative in halophyte, and we could introduce that signaling in glycophyte, because they do have all the the machinery, salt tolerance machinery, but it is like not expressed at that level, and that promptly. So I think that could be a good venue for the molecular biologists to impart salinity tolerance by learning from the halophytes. Yeah, this is what I believe. Yeah.
So thank you so much. Any other question? Okay. You have one as well? Yeah, we do have another one. So.
Thank you. A question to Dr. Krishna. Dr. Krishna, you have done excellent work, highly appreciated, high quality, state of the art.
Yes.
The question is how to reach to the farmer, how to transfer this technology to the farmers. This is my question because farmer is very poor, he has not reached to such high level technologies. So what if there is any gap, how to bridge the gap between the farmer and this high tech what you have presented? Thank you.
Thank you so much, Professor. Actually, this is a good question. And we are not asking farmers to run the model. What we will do is using the model output. That's what we are doing also. We make the dashboard. We make simple ICT tool, advisory tool, and then that we will offer to the farmer. They will not be running the model. In Australia, farmers, high-level farmers, they are running, but I am hearing. But our context, I have not seen, and this is not for the farmer. We need to do, for example, I make the result for 51 crops. What we are doing is now we are plotting it, we are making the dashboard, and we are making very simple tool, and then that may run in mobile app, and then that will go to the farmer. That's the reason that actually model is the real decision support tool. For farmers, it is a good decision support tool for researchers, of course, universities, students. Actually, just 2-3 weeks ago also, I was not knowing a lot of things about wheat in many regional countries. When I started to run, I learned a lot of new things. This is so good tool, freely available, and especially for the young generation students, I highly recommend that kind of things to use. And from using those outputs, they can make all is for to serve the farmer. Yeah, my point is farmer cannot run the model. From this, using this model output, we make a small, simple decision support tool, advisory tool. This is the planting data weight in that region. For example, that way. This is the fertilizer rate for this region of Tunisia. That way it goes. And inside it can run model. But actually, currently I'm making a small decision support tool also using those things already. Around 10 questions. When I was in ERI, I make one tool that required around many questions to answer, and then that did not fly. And my point now is a bit of less answering type of question we need, and then inside we can put algorithms. There can be remote sensing thing also can go. That model can run inside. That's how we do. For example, there are tools which has used inside model, but the farmer will not know that one there is model, they are running it, considering the climate, considering their soil, it is generating, they don't know. They just answer a few questions and then it goes. Yeah.
Thank you so much. Let's have one last question from Professor Ahmed.
Thanks, Butaina. My question is to Professor Abdelhamid first and to Hasna after. So for the Halophyte, I want just to understand if they can contribute to the carbon sequestration, and if the answer is yes, what kind of response we should expect. So if we have like some sequestration, in saline areas when we have like Halophyte, we'll have more carbon or less carbon, or at the level of the road system.
Yeah, thank you very much for your question. Yeah, the halophytes can also be used for these carbon sequestration, and especially in the coastal areas. Nowadays, there is a blue carbon economy, and blue carbon ecosystem is a kind of buzzword, and it also supports livelihood of a lot of the local tribes, fishermen, So yeah, in many parts of the world, different groups are working on utilizing halophytes for this carbon sequestration. We can even utilize them for the greenification of, for instance, while traveling from the Laayoune city to the campus, I observed that on many parts of this road, the dune was covering the road. So I believe that is a kind of pain. It will cost a lot of gas money and a lot of labor. So if halophytes, the sea is not that far, so many halophytes such as many halophytic grasses can be used to fix those dunes. They can be grown, so they will sequester carbon, they will improve the overall environment, it will add to the beautification, as well as it will save time and money and labor for this the road cover from the dunes. So yeah, that is a very a viable use for the halophytes. In addition, the halophytes can also be used for other than the carbon sequestration, as I mentioned in one of my slides, that they can even be used for as food. For example, quinoa is a very good example. Quinoa is a halophyte. It can be used to produce grains. Similarly, salicornia. There are a number of halophytes. Portulaca. There are a number of halophytes which can be used for different economic purposes. Yeah, and also and even that kind of work which actually she performed, ecological work, seasonal variation. That is also I believe is very important. Even it can contribute to food security in long run. By understanding the biochemical footprint of what is going on in plants under extremely scorching sunlight and extremely dry condition. One can do chemical engineering in future, even in the crop plants, to impart salt tolerance. So I think halophytes are going to stay in this in this circle, and they are going to contribute different in different ways for food security, as well as the carbon sequestration, as you mentioned. Yeah.
Thank you.
What is the question? Is it the same? The same for carbon sequestration. Actually, there is no study about Haloxylon salicornicum regarding carbon sequestration. But actually, what we have in the desert is inorganic carbon. We don't have organic carbon. And for breeders and agronomists, they need organic carbon. I think it's not useful for that.
Okay. Yeah.
But we don't have organic carbon in the sand.
No, but I mean, you have your plant, you have some microbial activity within the roots. You have some decomposition of the organic matter to get like organic carbon. So maybe you don't have like a high.
It's perennial, yeah.
Like all the plants have different capacities to sequestrate carbon. Either it's low or high, but we need maybe
actually
to do
I have
the
no
study.
idea about that.
I mean, even the biomass of the plant, you can sequester some carbon through photosynthesis.
Yeah, from the atmosphere. That's right.
That could be some
By
kind of
photosynthesis,
service plant also. That's the answer to the first question, just to tell you that you can find some application, ecological application for the plant. And from there, you can just think about how people, they can use that plant to generate maybe some revenue, or that was
some legumes, some crops,
some income, some money. Yeah. Yeah. Okay. Thank you very much, Hasna. Good luck with your work. Yeah.
Thank you.
But yeah, you should think about the application and the implication as well. Thank you.
Maybe.
Good luck.
Thank you.
Yeah.
Just to add carbon like to your soil analysis. That's
but
all.
I'm done, actually.
Okay, maybe for another PhD student.
Okay.
So one last question from our PhD student.
Wonderful experimentations, Professor Abdelhamid. I wanted to ask you about the graphs. You talked about priming, the concentrations, and I wanted to know if there is a relationship between the exposure, the duration of the exposure, and the concentration. Is there any correlation or relationship between the two?
Yeah, to save time, I did not include, the entire, data. So we initially performed kind of preliminary experiment, and we use, different concentrations as well as different exposure time for, priming, and we then selected like the optimal time as well as concentration as well as the chemical for the priming. So for priming to work, you have to have like optimal concentration. First, you have to select the appropriate chemical, the concentration, as well as the exposure time. So all these three are very important. Yeah.
Thank you so much.
So thank you so much. Thanks to all the presenters and also the participants. We were delighted to have you in this session, and thank you.
Assalamu alaikum. Good afternoon. Honorable guests, dear students, and research community, scientists, researchers, and colleagues. So we are at the end of this nice conference. We've got to enjoy our time during these 3 days, and this is the closing ceremony. So we're gonna start the closing ceremony by reading some take-home message that I collected from different researchers and scientists, and we summarize them in some few slides. So I will read this this recommendation based on the outputs from this conference. So the International Laayoune Forum on Biosaline Agriculture and Arid Land Agriculture has reaffirmed the urgency and the potential of transforming agriculture in saline and arid environment through science-driven, climate resilient, and locally adapted innovations. So the insights shared over the past 3 days highlight the critical intersection of water scarcity, soil degradation, and climate stress, and the innovative paths that we must, as researchers, to take and to secure food system for the future. Participants from across the globe have demonstrated that despite harsh constraints that our regions suffer from, biosaline and arid land agriculture holds an immense potential as a catalyst for sustainable food production. Effective collaboration among researchers, farmers, industry leaders, and policymakers is not merely beneficial, it is indispensable. This forum has served as a vital platform for cross-sectoral dialogue, the exchange of cutting-edge knowledge, and co-creation of actionable forward-looking strategies. The first part of this recommendation, as highlighted by one of our keynote speakers, Rashid Sarraj, that he mentioned that there is a need for our research for development for impact. This is really a good take-home message. So it means that our research should focus on the beneficiaries and the farmers, and farmers should be at the center of our research strategies. So delivery is required and mandatory for the large impact, needs to reach the unreached and touch the untouched. The research should focus also on farmers' needs, and farmers should be involved as the key stakeholders to benefit from the research and to adapt the solution. It means that our methodology, whatever we are doing in the science, should come from the problem that normally farmers are facing in their field. So this is the first key take-home message. The research should be aligned with the environment condition and oriented towards market demands. So we witnessed some presentation that stressed a lot on market demand. So without the market, our research will be just in the shelf and will not be valuable for the beneficiaries. The second category of this recommendation concerns the scaling up of salt-tolerant and genotypes. We have got a lot of presentation that are talking about improving crops and varieties, using a lot of techniques and method. So but first we need to invest in landscape level salinity mapping, because this is the base, that will show us actually the current situation of our land. And of course, to adopt real-time monitoring technology for better decision making. Also, we need to accelerate the breeding and deployment of salt-tolerant and drought-resilient crop varieties using both conventional and molecular tools. Exploring the potentialities of using wild and native varieties. We have been talking about the use of wild relatives. This is very important. This is like a good nature-based solution to improve resilience under changing climate. Also, there is a need to adopt the integrated management approach in our research and application. And that accounts actually for soil, plants, water, fertilizer, and energy. We need also to build resilient communities by providing them with the right information and data, with the right seeds, and also with the right tools to cope with harsh environment conditions. Also, researchers need to assess the impact of their research on the environment, not just doing science, but we need to assess, evaluate the impact at different levels, at the environmental level, the ecosystem, and also the socio-economy and sustainability concept. It is also recommended to encourage the integration of information technology, the new technologies like Internet of Things, AI, and other digital tools with the biological science. So we need to integrate this to the biological science and agronomy as well. Regarding the strengthening capacity building and farmers' engagement, so we need to simplify the finding actually, because we work a lot, but our science doesn't reach the farmers simply because they can't understand our language. So we need to simplify the findings to be easily understood and applied by the beneficiaries and extension services. So there's a lot of efforts how we can translate the science that we're producing to reach the farmers in a simple way. And I guess a lot of scientists are working on this aspect actually in their respective institutions. Capacity building should be strengthened by using mobile apps, for example, because now nobody in the globe, who doesn't have any smartphone. So this has become something very easy, and the information is easy to access. So we need to shape our extension and our information to be delivered to farmer in an app, mobile, for example, social media, and farmers' open day to accelerate the knowledge transfer and the adoption of our technologies. There is also an urgent need to develop seed production system because we are talking a lot about new varieties, new crops, but from our experience, actually, we have a gap in the seed production system. A lot of farmers got interested in some crops or varieties, but actually, we don't have the capacity or we don't have this seed production system, to support them. So there is a need for this kind of system. And also, we need to make the seeds available at affordable price for the farmers. There is also a need to integrate biosaline agriculture into national policies. So in many countries, biosaline agriculture is not yet recognized, although we have a big challenge and problem in salinity management. So we need, from the government side, they should mainstream biosaline agriculture into national and food security and also climate adaptation strategies. Because it's a fact, it's a reality, and we need to act before that this problem become bigger and bigger. The public-private partnership also need to enhance our research development. And we have here in Morocco a very good example for the private companies that are investing a lot in the research and development. Finally, promoting international and interdisciplinary collaboration is also a very important recommendation by this conference. Strengthening networks and events such as LAFOBA, for example, INSAS, networks and other to facilitate the ongoing exchange of knowledge, data and germplasm across countries and institutions. Also, we need to encourage joint projects that integrate agronomy, hydrology, socioeconomic, and policy for a holistic solution. Because what we are facing today, the salinity or the drought, They don't need only one unique solution, but they need an integrated solution from different dimensions. So that's it for the recommendation. And now I think we have another chapter for this closing ceremony. So as you know that in our research, we work a lot with farmers. So this is the philosophy that we have at UM6P. And in each edition, we'd like to recognize, some farmers that they participate in our trials, in our research. I'm calling a few farmers that participated with us in our research to come to the stage and to give them some, trophy just as a way for their recognition. I don't know if we have Baqada كاين هنا محمد لامين كاينش next جيد السباعي كاين تفضل I'm inviting Sibou Hsini, who is our actually research director at the College of Agriculture and Environmental Science at UM6. Mustafa Musta'in is a farmer working with us on the upscaling of blue panicum and other forages. He has got his land affected by salinity, and he's one of the good example actually of what we call champions farmers, because he's not adopting the solution, but he's helping us to reach other farmers. I'm inviting Dr. Ragab. Please come to the stage to deliver the Next, Si Mohammed Ayash is also one of the farmers that collaborating with us in the region. I'm inviting Si Hijri. Next from Arhamna region, and by the way, they are not from the same region, as we are working at Asari, working in other region in north of Morocco that are suffering from the same salinity problem. So I'm calling Moulay Abdouahed Idrisi from Hudaya Invest Company. Mark. So next, and the last one, they have a farm in the middle of nowhere in the desert, in Boujdour area. And just to give you an example that inside the desert, they can actually have crops and they can cultivate. They have a lot of challenges there. They have winds, they have salinity, but they are actually, maintaining their productivity and trying to do their best to make that area green. I'm calling Sidi Ahmed Khalifi from Boujdour area. Shabir. Shabir. And now actually come the moment of truth. I mean, the chapter that everybody is waiting for. It's the awards for junior researcher for PhD students. And the last moment we were making, scores and calculation. And finally, we got the result of the two best prizes for oral presentation and for posters. And we have three places, first and second and third. And please give me the first we start by the best oral presentation. First, please. So I'm calling first on the stage Mr. Douali of the region of Laayoune Saqi al-Hamra to deliver the prize. Please, Mr. Douali. So the first or the best, we start with the first or the last one? What do you think? Last one. Sorry. Give me the last one, please. So the third place for the category of best oral, please not poster. Sorry guys, so I'm calling Fadwa Mekawi for the third place of the best oral presentation. The second place for the best oral presentation goes to Sheima Rami. And now we announce the winner of this category. The first place, best oral presentation, Bushra Darqawi. So we still have the best poster awards. So we start by the third place, Omar Najari. Finally, we have a man here. A boy. And now the second place goes to Sofia Baha. And now we move to the best poster award first place that goes to Lamia Daoudi. So last but not least, I'm calling our director of the African Institute for Research in Sustainable Agriculture, ASARI, and the leading project lead of the UM6 University in Laayoune region, Mr. Mfaddal Kwisni, to give the closing remark of this conference. Thank you.
Thank you, Sir Aziz. Mr. Douali of Laayoune Saqiya Al-Hamra region, distinguished participants, delegates, partners, colleagues, and friends, Assalamu alaikum wa rahmatullahi ta'ala wa barakatuh. Organizing such an international conference on biosaline and arid land agriculture here in Laayoune at ASARI is one of local knowledge combined with innovation in agronomy, plant physiology, genetics, microbiology, and all the other aspects that you have discussed for the last 3 days could drive sustainable environment in improvement, in arid land farming, ensuring food security and ecosystem preservation. As a third attempt through this third edition,
we tried through this conference to offer farmers, cooperatives, and socio-economic scientists the opportunity to meet, learn from each other, and build collaboration. As you have witnessed, the program of this third edition was carefully set to cover the topics of major importance and address the issue that impacts the productivity and livelihood of the farming communities living in arid zones. Through your great contribution, we have shown that sustainable agriculture solutions are possible, even in the harsh conditions. You have shown to all of us that we have the tools, whether it is through the development of salt-tolerant crops, integrated soil and water management systems, conservation agriculture, or leveraging indigenous knowledge for scaling innovation through farming training, field demonstrations, and inclusive technology transfer. I would like to take this opportunity to thank you all for your contribution, dear speakers, presenters, participants, and partners, for contributing to this success of this conference. A special thanks is extended to Mr. Douali of the region Laayoune Saqi Al-Hamra for his continuous and sustainable support. We are always very honored and delighted to have him with us. So thank you again, Mr. Douali. We are really very honored to have you exchanging with the scientific community and the local farmers as well. A heartfelt thanks is also extended to our sponsors, Phosboucraa Foundation, OCP Phosboucraa, and UMCXP, whose generous support has made this event possible. I would like to express my gratitude to ICARDA, our partner that joined us in this the organization of this third edition. I would like to thank the members of the organizing and scientific committee for their great contribution and hard work. Of course, I cannot forget to thank the Asari facility team and the health center, the HSQ and communication teams, as well as the IT for their contribution. I will probably specify that our colleagues from communication, Ben Guerir, and IT team from UMCXP Ben Guerir came to support us in this event and to make it successful. A special thank also extended to the CLIC team, Mariam, Shamsuddin, and their colleagues for their support, the technical support and the logistics support. And before I conclude, I would like to warmly thank, but also congratulate Professor Aziz Hirish and his team for their contribution and the well organization of this event. Thank you very much, Aziz, and congratulations. Well done. A specific thanks go to the PhD students, because when I saw who was supporting Aziz and young PhD students were contacting all of you, making the program together, the presentations, helping the scientific and organizing committee, I said we have, a new generation coming of good researchers and good professors, inshallah. I hope that you have spent good time with us for the last 3 days. The coming edition is coming in 3 years. I hope also that you enjoy all what you have seen. The foods, the people, the region, the area, but also the scientific content that we shared with us for the last 3 days. I hope you had a fruitful, productive, and enjoyable scientific discussion and a very pleasant stay in Laayoune. in the southern region of the Kingdom of Morocco. And we are looking forward to seeing you all with your friends that could not make it this time for the upcoming edition, the fourth one, that will be organized 3 years from now, inshallah. So don't hesitate to share the information and invite your colleagues to join us in the future, inshallah. Before I close this session, I would like probably to invite to the stage the scientific and organizing committee to take a picture with Sayyid Abdulaziz Hirish to really memorize it, because it has been wonderful effort by all of you. Please come to the stage. And while they will be preparing here, I would like to invite the speakers or all the participants, if you have time, of course. I know that the schedule was quite tight. We didn't get time to show you our laboratories and research infrastructure. So at the end, you can just stay at the lobby here, and we'll take you to the research laboratories if you want to have an idea about our research infrastructure. Thank you again.
So please don't leave. Just I have an announcement, a quick announcement. So I mean, for those who are extending their stay here in Laayoune for tomorrow, or are leaving maybe tomorrow night, we are organizing with Professor Ragab a workshop about the salt-made model. So it's a crop model. So anyone who is interested to join tomorrow morning, we're gonna start at 9 o'clock. The pickup will be at 9 o'clock, at 8:30 from Laayoune. So we're gonna have our bus. So take, the people from Laayoune to join a workshop here. So it will be about the salt-made model, facilitated by Professor Ragab. And I think there is another announcement by Professor Bartali, who is organizing a conference. Bartali, do you want to come and talk about the conference? Just a few minutes. And then probably in the meantime, since I don't know if you checked out already from the hotel, because the buses will go directly to the airport, just to avoid any delay in the check-in process. So the flight is planned 8:50, I guess, right? 8:50. So you should be there at least like 7 o'clock. You have enough time to do the process. So I guess the bus will depart at 6:15. We have like maximum 20, 25, 30 minutes to reach there. Okay, so let's say 6:20 they depart sharp. Okay, so everybody should be in the bus 6:20 to be there at before 7 o'clock. Okay, so Bartali, you want to?
Thank you very much, Professor Harish, and congratulations. My name is Bartali Al-Hussein, professor from Institut Agronomique et Vétérinaire Hassan de Rabat, Agriculture Engineering. And I'm also Vice President of the Moroccan Committee of Irrigation and Drainage. I want to welcome you all to attend the coming conference. It's a Pan-African conference in Fez in October 2025, from 22 to 24 October, on the topic of water management for sustainable and resilient agriculture in Africa. So I thought, because of this prestigious community, we can still benefit from your input, given that we are here at the African Sustainable Agriculture Research Institute, and we are organizing this in collaboration with several partners, including ICID, for which the honorary president, Dr. Ragab, is here. We are also having IMI, the International Water Management Institute, and Water Witness from UK, and the French National Committee of Irrigation and Drainage. In this conference, we promise to give you the opportunity to cover a number of subtopics to make sure that there is a lot to do to mobilize water and use it properly, from conventional resources, non-conventional, the biosaline agriculture that we have seen here. There will be two visits, one to the major agricultural projects in Sais, where Moroccan ministry is trying to save the water table of Sais with irrigation water coming from the barrage, the dam of Mdez, and also one going to visit to the wastewater treatment plant of Fez, which governs the river of Subu, from which Morocco is taking water to another watersheds. So I invite you, please, to look at the website of the conference. I have put some announce sheet in the conference, but I will be ready to answer any questions for those who would like to contact me. Thank you so much.
Thank you. And now, if you like to do a lab visit, I mean, for our facility, for those who didn't do it yet, so I'm inviting you to gather like in the reception area. There will be our PhD student to guide you there if you are interested, of course. Since we still have some time, we have like 30 minutes, 30 minutes to go. And that's it. So finally, I would like to thank you a lot for the interest, for submitting your abstract, for presenting your presentation. An extended gratitude goes to our prominent keynote speakers. Thank you a lot for accepting our invitation, and also for all the students that joined the conference. We wish you all the success in your career. And as you know, maybe if, your organization interested to join the conference, probably for the next edition that will be in 3 years, so please come, feel free to contact me to be a partner or co-organizer, so we are open to make it probably the next edition bigger and bigger. So we're gonna aim probably for more than 500 participants, and we are inviting all of you, your organization, to join us for the next edition. Thank you so much.